Hello, my name is Sebastián Hernandez. I'm a third semester medicine student, and we're going to discuss about this article for the assignment of molecular biology. We're going to discuss a lot about this later, but first, we've got to talk about one of the main axes of the article, that is breast cancer. Breast cancer refers to the pathological growth of cells around the breast area that refuse or lose the ability to die, consuming so much energy and refusing to die, leading the host to its death. The other axis of the article is the KTM4A signaling pathway, which is a protein in charge of methylation of histone H3K9, which leads to DNA repair, cell growth, and proliferation, basically creating a tumor because this protein helps cells that are supposed to die, cancerogenic cells, to keep repairing their DNA. Well, the general objective of the article is evaluate epigenetic regulator KTM4A activating NOX1 NICD dependent signaling to drive tumorigenesis and metastasis in breast cancer. This is just what we discussed before with the KTM4A signaling pathway. Materials and methods. The first method we've got is the immunohistochemical staining. Basically, 50 samples were extracted between 2014 and 2020 and then analyzed with G-Division 3 staining kit. Then the staining samples were classified into non-weak, moderate, and strong, or 0, 1, 2, and 3, depending on the staining intensity and the cover area. The next method is RNA isolation and quantitative PCR. RNA was extracted from the cells previously cultured and then transcribed to cDNA via reverse transcriptase, and PCR was performed. PCR, or polymer chain reaction, is a method that helps provide information of quantification of how much RNA was transcribed for some certain specific proteins. In our case, is KTM4A, NOX1, and its downstream genes. Next method is Western blot analysis. First, they perform electrophoresis, which is putting proteins into a gel and then electrifying it. The electrification, or basically getting electrical current into a cathode in an anode, helps the proteins arrange into protein bands according to their weight, their charge, and their size. The next one, after the electrophoresis, a Western blot was performed, which is basically a antibody-antigen relation. Antibodies were specific for proteins important for the study were marked for these proteins exactly. After the electrophoresis was performed. The next is qPCR assessment. Basically, proteins were bound via cross-linking to certain specific parts of the corresponding DNA. Then everything that wasn't bound to a protein was fragmented and then again marked with antibodies. The higher weight antibodies marked proteins with DNA was precipitated for later DNA purification, leaving the DNA only. And once again, PCR was performed to help elucidate how much RNA was being synthesized for these certain specific proteins. For the results, first we've got the KTM4A silencer RNA. We know about two specific kinds of silencer RNA for KTM4A human. Basically, we've got MCF7 culture, which we're going to discuss about later, a control, which is basically the cells without any manipulation, and then the knockdowns, which suppress KTM4A. And we see that this worked because KTM4A protein band was a lot more thicker and darker on the control group than on the knockdown cells. This relates directly to the third result, which is the signaling pathway. In the knockdown cells, we see that there is no KTM4A being expressed. The NOX1 protein is less expressed. The band on the knockdown cells is a lot lighter. It's not only lighter but thinner compared to the control group. And then we've got the low-high classification. We basically got cultures, all these culture names, and classified them on low and high depending on the thickness of the KTM4A blood line. In the first three, we see that they are lighter and thinner lines than the last three. Next we've got KDM4A knockdown and overexpression. We've got MCF7, which is a culture and its control group, and then the knockdowns. We see that in knockdown, cell proliferation and invasion in tissue was a lot less compared to the control group with KDM4A high. And then we've got BT20, which is basically the same but in opposite. We've got a control group which is KDM4A low, and the other group which is purposely overexpressed KDM4A, and we see that cell tissue invasion was a lot higher. Next we've got Notch1 inhibition could suppress downstream genes. This refers that in these bloodlines where there isn't a knockdown for KDM4A but for Notch1 instead, where the Notch1 line is a lot thinner and a lot lighter than the control group at KDM4A positive, we see that these downstream genes are a little bit lower, have a little bit lower expression than the control group and a lot lower than the KDM4A positive. Next we've got KDM4A increased cell viability and proliferation. Basically, once again, Notch1 knockout and control show that compared to KDM4A, cells live a lot less time than KDM4A positive. Well, the discussion, we've got these three statements which all relate to KDM4A overexpression, Notch1 activation, and histone demethylase from these authors, and according to this text, everyone agrees. And finally, we've got the conclusions, which are asserting molecular pathways in pathological events could help provide therapeutic targets such as KDM4A or Notch1. And learning about the tissue behavior behind protein overexpression and knockdown might help establish the reason to certain pathologies and develop more specific and efficient treatments.